So before we start talking about the editing, maybe let's think about why we talk about gene editing, right? So I want you to think about DNA as really the blueprint for all of life, right? DNA is the set of instructions, and it's going to be encoded into all of these different building blocks that's going to make up you as a human or a plant or a microbe, a virus, a bacteria, everything that you can think of, or almost everything that you can think of that we would classify as alive, the instructions for that life is encoded in DNA, right? And I want to spend a minute just thinking about this quote from this really wonderful book called The Gene and An Intimate History by Siddhartha Mukherjee. So I'm going to read it out loud, and I think it's just one of those really powerful concepts. If you haven't already read this book, I highly recommend it. It's absolutely wonderful. So he says, the atom, the bite, and the gene provide fundamentally new scientific and technological understandings of their respective systems. You cannot explain the behavior of matter, why gold gleams, why hydrogen combusts with oxygen without invoking the atomic nature of matter, nor can you understand the complexities of computing, the nature of algorithms, or the storage or corruption of data without comprehending the structural anatomy of digitized information. Alchemy could not become chemistry until its fundamental units were discovered, a 19th century scientist wrote. By the same token, as I argue in this book, it is impossible to understand organismal and cellular biology, or evolution, or human pathology, behavior, temperament, illness, race, and the identity or fate without first reckoning with the concept of the gene. And so I think this is where I'm coming from in this talk, is where do we start breaking down the components of what makes us us, what makes life life, and what happens when we decide to not only read and understand, but also to change these components. And so why should we talk about this now? Like, why is it so important to talk about these technologies in the current moment? So for the most part, for the most of human history, we haven't really known anything about DNA, anything about our genetic information or the codes that make ourselves ourselves and that make us us. And to give you some context, the discovery of the DNA structure. So when we first realized what our DNA actually looks like, that article was published in 1953. And so really in this short span of time from 1953 to now, really in a single lifespan, we have gone from not only understanding the structure of DNA, but then to moving forward and being able to read it. In many cases, being able to understand what the code means, and now most recently in the last decade or so, being actually able to rewrite it, to make edits and really specific edits. And so it's this really powerful, powerful, revolutionary change, but it's also been incredibly quick. And whenever change and such powerful change comes so incredibly quickly, I think it is on us to take pause and reflect before we actually start using that power. And so I started my talk by telling you about CRISPR-Cas9 gene editing and how powerfully it had changed the ways in which I was able to work in the lab. So let's think about maybe taking a step back and asking, well, what is this CRISPR-Cas9 gene editing? Where does it come from? And so it's originally really a bacterial immune system. And I'm a really visual person, so that's primarily the tool that I'll be using to communicate. So let's imagine this is a bacteria and it is infected by a virus. What the virus is doing here is it's injecting that yellow stuff into the bacteria. That is the viral DNA. It's the virus's genetic information. And the end goal for the virus is to take over the bacteria, to turn that bacteria into the ultimate virus-making factory. And so what the bacteria does as it fights off this viral infection is not only does it aim to fight it off, but it also reads this genetic code and it actually cuts out short snippets. So it essentially takes short snapshots of that genetic information and it stores them in a database. So it just has, over the long term, if it survives that viral infection, now a database of previous viruses. And so the next time, if a similar virus comes along, that database is going to recognize, I see you. I've been infected by you before. I'm not doing this again. And so it has this really efficient pair of molecular scissors, and it's just going to snip, snip, cut it off. No viral infection. We're all good, right? And this will happen over and over again. As many viruses that will come, as long as they share the same DNA sequence, the same genetic code, they'll just be snipped off, and the bacteria is now protected. And so in looking at this, what I want you to take away from this is that this system has two components. So it has what we call the CRISPR component. That's that mugshot. It's that series of short little sequences that give it the identity, that really target the ammunition. And then it has this second component, or the Cas9 component, which is that molecular scissor. So it's going to, once targeted, cut genetic information, cut the DNA into two. Okay? And now, intuitively, you might realize how powerful this technology is. Because if we can just replace that mugshot, if instead of targeting it in bacteria against specific viruses, we can put our target of choice in there, we now have this really powerful tool where we have these incredible specific molecular scissors that now we can target at pretty much whatever we want. And that's CRISPR-Cas9. And so maybe you're thinking, well, you said you were doing gene editing before. Why is CRISPR-Cas9 so special? Why was it so revolutionary? Here's a few reasons why. One, as we just spoke about, it's incredibly easy to program. It's just a matter of changing this short bit of DNA that targets it to the appropriate location. And we're incredibly good at making short bits of DNA in the lab. Just chemists figured it out a long time ago. So that is one reason why it's so very revolutionary. But the second reason is that it's also really accurate. So when we say target, we mean target. It will target just that really specific region and make a cut. It's also really fast, right? Remember, 2-3 years to 2-3 weeks. So that type of change is what we're talking about. And I'm just going to say it, I'm not the most brilliant researcher out there. I'm a good researcher. People can probably do it even in a shorter time, right? It's also really cheap. It's gone from tens of thousands of dollars to a few $100s. And to me, the most important, it's widely applicable. There isn't a species, there isn't an organism, there isn't a cell type that I have read about that CRISPR-Cas9 has not worked in. It's just universally an editing tool. And so that brings incredible excitement in the scientific field. So what should we be worried about with CRISPR-Cas9? And really, if you think about it, exactly the same things. It's incredibly easy to program. It's accurate, fast, cheap, and can be applied to anything. So while that's really exciting, it's also, to me, a chronic worrier, incredibly worrying. What are we going to do with this? Who's going to have access to it, and what are they going to do with it? If you're thinking I'm exaggerating about quite how cheap or accessible it is, you can buy a DIY bacterial genetic engineering kit for $170 online, and they will ship it straight to your home. This exists. So what can we really do with CRISPR if we want to focus on the more hopeful side of things? We can use it to develop drugs. We can use it to study animal models and really understand pathogenesis and disease systems. We can perform gene surgery. You can simply edit out a faulty gene and edit back in the correct version of the gene and cure genetic disorders. We can make clean bioengineered fuel. We can make food more pest resistant. We can make higher yield food. It's just as many possibilities as you can think of. Those possibilities are out there. And so maybe let's just talk about a very, very few of these. I'll give you a couple of examples from maybe each different field. So let's start with biotechnology. When we talk about biotechnology, maybe we're talking agriculture, farming, and how could we apply gene editing to to this practice. And so one example of a project that is currently being pursued is a project where they are trying to genetically edit dairy cattle to make them hornless. So if you don't already know this, certain cattle breeds are really specifically bred over decades of domestication to be really suitable as dairy cattle. They have high yield of milk, really good quality milk, and so on. And then other breeds of cattle are really suitable for their meat. And so as it turns out, through this decade-long breeding process, what we ended up with was dairy cattle have horns naturally, and meat cattle breeds just naturally do not have horns. They have a mutation in that gene that causes the horns not to develop. And so maybe we think that why resort to gene editing? It seems like picky and new and difficult to do. Why don't we just do the traditional breeding method? So you breed dairy and meat cattle together, and then you find an offspring that has no horns and you're all set. Except you're not really all set, because now you have an offspring that has 50% dairy genes and 50% meat genes. And now it doesn't make any good dairy, and it's also not good for meat. Yeah. And so you'd have to do this breeding process for 10, 20 years to eventually get all dairy genes except for that horn gene. So it's incredibly tedious. It also takes an incredibly long time. And now you also might be thinking, who cares about horns? Why should we care about this? But it's a very big monetary issue. For me, more importantly, it is an animal welfare issue. Animals, when they're kept on farms and when they do have horns, can harm each other. They can also harm the farm hands. So currently, the way that we deal with this is we dehorn the dairy cattle. And so the argument being made here is if we can perform this really quick and efficient gene surgery, we can have these breeds that are naturally hornless. We are not creating new genetic information that didn't already exist. We're just taking it from one breed and splicing it simply into this breed. That's the argument being made there. Another example, tomato plants, very, very big crops, right, all over the world. Tomatoes specifically are resistant to a virus called the leaf curl virus that infects the leaves, and it causes them to wither up and die. And it's also incredibly infectious. So the first image there is from WikiHow. It's how do you deal with leaf curl virus? And the literal instructions are you put a bag around it, you tie up the bag, and then you dump the plant. That's how you deal with that. And so the argument again being made here is that is wasteful, and it is sad, and it is also resource intensive, especially if you're a big farmer. And so if we can cultivate breeds that are resistant to the virus, that would save a lot of time. It would also increase the yield. Another big field that we can think of, very dear to my heart, ecosystems, ecology. My master's was in zoology. I spent a lot of time doing field work. And so something that I'm really passionate about is conservation biology. Don't come as a surprise to you that humans have messed up the earth in very many ways. And now very many animal species are at the verge of extinction. And one might argue that if we can, then it may be our ethical responsibility to do something to fix this mess that we have created. The first graph that you see here is an estimated global extinction risk based on the IUCN red list that they put out. And it's a little bit small, but what you see, each bar is not one species, but it's entire groups of organisms. So we're talking like amphibians and conifers and mammals. And in these groups, what you're looking on that x-axis is the percent of that group that is currently at least some risk of extinction. So it's just these wide swaths of living beings that are currently at extinction risks. And we can use gene editing technologies to potentially introduce new genetic variation, to take those small populations that we have left alive and make them healthier. We can maybe make them resistant to climate change. So there's a whole host of different applications that we can use if we choose to. Beyond conservation biology, we can also use gene editing to control vectors and disease transmission. An example that you might think of is Lyme disease or malaria, both of which have those insect vectors. So with Lyme disease, you have mice that carry ticks that may be infected with the Lyme disease pathogen. And as the ticks are transferred to humans, the humans may be infected with Lyme disease as well. And so the thought or the project being proposed here is that we could do it one of two ways. One, we could engineer ticks to be resistant to the pathogen itself, or we could engineer mice to be resistant to those ticks. If the ticks don't have a host, they don't get to survive, and then they don't get to infect humans, right? And at first that might seem like a no-brainer, like, yeah, if we can save people the suffering of Lyme disease and the suffering of malaria, then we should, right? Except that we're talking now about large-scale ecosystems engineering. So let's take a closer look, because I sneakily put in this picture of this little mosquito population, and it has these little red and blue mosquitoes, and I'm like, yeah, gene edit, awesome, right? But is it quite that awesome? Because let's take a step back and see what they're proposing, right? So in general, if I was to say, I'm going to engineer some mosquitoes and I'm going to release them into the wild, and then we'll see what happens. This is the normal inheritance, is what we'd expect to see. The red mosquito is engineered. It will sometimes pass on the gene to some of its offspring, in general, about to 50% of its offspring, right? So every time you see that curve split, you have two offspring, around 50% of the time, one of the offspring will get it. And so at the end of 2, 3, 4 generations in that large population, I released one gene edited mosquito. And after a few generations, you have a few gene edited mosquitoes out there. Now, is that questionable? Should we talk about that? Sure. But that isn't even the worry here. Let's look at the other end of the spectrum. The other proposal that we have is what we call a gene drive. So now we're not just talking about I'll engineer resistance in the lab and then release those mosquitoes that are resistant to the malarial parasite. Instead, what we're saying is I'm going to make that mosquito resistant to malaria. And then within that mosquito, I'm going to embed that entire CRISPR-Cas9 system. Essentially, what that does is it gives the molecular scissors and the target site into that mosquito that you are going to release into the wild. The gene editing is not just there. The gene editing tool is also embedded in there. So now any progeny that are made, any offspring that are made, every single one of those is going to inherit that scissor and the target. And every single one of those offspring is going to become genetically engineered as well. It's going to become resistant. And that, again, makes me, in some ways, incredibly hopeful, because so many people have suffered and died from these diseases. But on the other hand, it really alarms me, because now we're talking engineering at a population level, and we're planning in 2 or 3 generations that the entire population will be engineered. And now very clever scientists have come up with very clever plans, far smarter than me, far more experienced than me. And they talk about experimenting on islands. So any edited populations will be kept to those islands. And they will not spread, across the entire world, except that we know that in every instance that we can think of before, stuff on islands never actually remains on the island. It always escapes to the mainland. So is that really a great plan? I don't know. What I do really find hopeful about some of these proposals is those same very clever scientists have also proposed a radically new way of doing science. Because this is not science where four people in a lab or 4 politicians at the state capitol can sit or should sit and make decisions. Because any decisions that are made will not only affect those projects, but essentially will affect the entire world and will certainly affect that entire local community. So the thought being put out here is we will put our proposal to the local community, to that island community, if that's what we're thinking about. And as a community, they can decide whether they want to have this proposal enacted or not. And so it's really about educating and talking and bringing in people with so many different perspectives. And that's really why I was so happy to come here and talk today, because this is not a topic from which we can opt out. It is going to happen. So the only option that we have is, are you going to educate yourself? Are you going to educate each other? And are you going to be a part of this conversation? Or is it going to be a parallel conversation where others are making the decision? Those are really the choices that we have. And so, is it really that simple? I know we've talked about some complexities with gene editing, specifically with gene drives, but even beyond gene drives, even if we're talking simple gene editing, well, who owns the technology? How much is it going to cost? Can we predict the direct and indirect effects? So if we have gene edited mice, how is that going to affect the other levels of the biome, the other levels of the ecosystem, other organisms around them? Where are we going to test our approach? And why do we choose those specific sites? The questions that you might ask is, once those edited organisms are out there, they're just really out there, and it's going to be incredibly hard to put that back in the box, if you will, right? Once it's unpacked, it's unpacked. So how do we make decisions? Who gets to make decisions? And what are our contingency plans? What happens when things go wrong? Because at some point, things will go wrong. And then what's going to happen, right? And so maybe the next and really final application I wanted to talk to you all about is medicine. And so this is really where we see a lot of hopeful progress with gene editing. Gene editing has been used to discover new drugs. This is very similar to what I was doing in the lab. It's also one of the least controversial uses of this technology. You're using it in the lab to find potential genetic gene targets, and then you're going to use traditional drugs to actually target those. So probably nobody's really going to argue that this is in any way really controversial. We could also see CRISPR as being the next antibiotic, right? And this, again, should be pretty intuitive because bacteria use it as an immune system, as a defense against viruses. But we can see turning it on those bacteria themselves and then targeting the bacteria. So that's another proposed use. What's really exciting as well is the proposed use of gene editing is in correcting those genetic mutations that might arise. So maybe in correcting mutations that lead to uncontrolled cell growth or cancer, that would be a straight cure for cancer, provided, of course, that we knew what mutations we should be targeting. In the same way, genetic disorders like sickle cell anemia, cystic fibrosis, genetic disorders that previously were a death sentence a lot of the time, right? That is what those disorders were. We can now see ourselves not only treating and improving the quality of life, but just straight up curing. And that is incredibly powerful. And so is it really that simple? Because if we want to think about it, and really the way I've pitched it so far is you change the gene, and so you change the genetic information, and that changes the building blocks. So that changes the body or the observable characteristic, right? You see a gene that's broken, it causes cystic fibrosis. You fix it, you now don't have cystic fibrosis. And that works that way sometime. But rather than thinking of genetics as this linear process, and most often when we advertise genetics, this is the picture you see, right? One single DNA strand, one nice little protein, pretty little box, everything's awesome. But really what you want to think about is this. And this is a genetic pathway for one aspect of one type of ovarian cancer. And so imagine yourself now trying to do gene surgery, and you're tweaking one little box and you have no idea what it's going to do to all of these other little boxes. And again, one pathway and one cell type of one type of cancer. That's the level of complexity that we're talking about. So really, this is the can we question, and the answer is sometimes. And even if we ignore the complexities in genetics, I really want you to think about, if I were to ask you what makes you, you wouldn't talk about one gene. You wouldn't even talk about multiple genes, right? So when we talk about all of these different complex networks, so all of those red and blue and green and yellow boxes, all of those genes interacting together in tandem, they all fit in in this new model that I just put up into that small little box called your genome. And then your genome, your set of genetic information exists within a cell, that exists within an organ, that exists within different systems. And all of those different layers interact with each other in really complex ways. And even that is me simplifying, because I haven't really counted for the environment. I haven't counted for your individual experiences, for your social and physical environments, for social structure and culture and politics and economy and ecology. And so if we really want to make predictable edits, we have to look at this system as a whole, as opposed to trying to break it up into little boxes and studying just one box at a time. So that's really the complexity that we are talking about, especially with human applications or ecosystems, those higher level applications that we talk about. And so the ways that we may think about human genetic engineering, we think about it in one of two ways. So you can choose to make somatic edits. Think about somatic edits as if I'm trying to, for example, cure somebody's cancer, I am only going to target those cancer cells. And those edits will only change those specific cells. They will not be passed on to any other cells in that person's body. And they will not be passed on to any offspring that that person has. Right? So that's somatic editing. Versus germline editing, where I'm changing all of the cells of the body, including potential sex cells, those sperm and egg cells, and those changes would be passed on to future generations, right? So that's one really hard line that we think of when we think about human gene editing. And for the most part, we agree that we should stay on one side of that line for now, until we have more scientific information and until we have more conversation about whether we really should cross that line. We should probably stay on that somatic side, because that way any changes that you make only affect that one individual and not all future generations. The other line that we might think of is this line between treatment or cure of diseases or disorders versus enhancement. And at first, that seems maybe like an easier question to tackle, right? We should probably just stick to treatments and cures. That's what's important. And maybe in some cases it is. So if we're talking cancer, we're probably only going to cure that one person who has the genetic mutations on cancer. And that is clearly a treatment that is going to save that person's life. So put it in this box right here. And if we think of cystic fibrosis, again, pretty clear here that it is a treatment or a cure. One might argue, well, we also want to save future generations. So maybe we put it kind of on this line here, where I'd argue we should still stick to somatic editing right now, but eventually in future as a society, we might decide to cross that line. So we'll put it there for now. But as you start thinking about all of the different pieces that make us us and all of the different things that we can come up with here, you'll realize that it isn't really a very clear distinction. So if I bring up, for example, things that may not clearly or cleanly fit in this box, things like eyesight, I wear these incredibly, considering how much they allow me to do, these incredibly cheap little pieces of enhancement that allow me to see and do what I love best, read. The reason I can't see very well is a genetic mutation, likely, that predisposes me to have poor eyesight. And you might argue that, well, that disrupts the quality of my life, and I should be allowed to correct that. And even though it's a genetic disorder, if you will, that's causing that propensity for poor eyesight, maybe the easiest fix is the glasses and not the gene surgery. So there's that. But there's also other subtleties. So let's consider human height as an example. And let's take one simple case study to really end the talk with, and to really focus on how these lines between treatment and cure are really blurry and not really fixed hard lines in the sand. So let's consider the case of David, who's a preschooler, who's the shortest child in preschool. And he's been the shortest child by 3 standard deviations below the average. So very, very short, the shortest end of the spectrum. Does have parents, biological parents that are also on the short side, but maybe not on the shortest end of the spectrum. So they're very worried, and they decide to test whether the short stature is due to some type of underlying pathology, which it could be in some cases. Luckily, the tests come back negative. But now David's parents read online that now human growth hormone therapy is safe and effective for children like David and may increase height. And they asked the doctor if they could opt into that treatment. And so the doctor says, well, insurance will likely not pay because it's not a diagnosed medical treatment with a specific cause. It's what we call idiopathic, unknown cause. We're not really sure why the child is short. And so the parents say, all right, well, we see it as an investment in David's future. And it may be even more important than something like a private school education, because socially, height is very, very important. And if you're thinking about, is height really important? Maybe the doctor's gonna push back a little bit and say, well, you learn to live with who you are. And the parents are gonna say, well, here are a few studies that we found. I'm just going to say here, all of these studies have fairly small sample sizes, but there are multiple studies here. So I'm going to leave it to you to make your own conclusions. In this first study, 140 recruiters are asked to choose between equally qualified hypothetical job candidates. The difference between the two applications is really just the height, 6'1" versus 5'5", and 72% of the recruiters prefer the tall candidate. And only about 30% of them are even able to acknowledge that they have equal qualifications. So it's that implicit bias that we have. We don't even realize what we're doing when we're doing it. There's another study that looked at a few hundred library science graduates, and they compared salaries between groups of individuals that were either taller or shorter than 6'1". And the average difference in starting salary when they divided up into tall versus short was 3 times more than when they divided up into more or less qualified candidates. So there's that. And again, there's a study of the Air Force where they look at people who are 20 years into the Air Force, and people who are shorter on average earn $2,500 less per year than those who are taller. Maybe these are all just correlations, maybe they aren't. But this is these studies are out there. There are multiple studies and they're replicated in multiple different contexts. And so now you see, if you just switch this human hormone, growth hormone therapy with gene editing, where that line really gets blurred. So maybe what I want to end with today is to leave you with even more questions. So really, just like in the example with height, well, who decides what we can and what we should edit? Who owns the technology? Can we predict the direct and indirect effects of gene editing? What happens if some people are able to access it, others aren't? When and where are we going to test our approach? Who are going to be the test candidates and how are they going to be affected? What are our contingency plans? Who is responsible for mistakes? And finally, who will have access? One final thing to leave you thinking, this isn't a CRISPR-based technology, but we do have certain FDA-approved gene editing therapies out there, already approved. One of the first ones that was approved was for a congenital blindness. It's a really rare form of blindness, and it costs close to $1 million. The company really generously has offered to allow people to pay in installments and has given a money-back guarantee. It's this whole new world of the ways in which we think about drugs. And on that, maybe not so happy note, I'm going to end the talk and ask you for questions.
Thank you, Dr. Masani. So now we're going to move into our question and answers. We already have a couple in the box, so I'll start with those. And if you have any questions as we go along, please put those in the chat, or you can raise your hand and I can call on you. So the first question comes from Bob Miller, and the question is, doesn't the gene drive method mean that any edit is reversible?
Yes and no. So yes, we can keep putting in more and more target sequences and editing changes. They've actually also built a self-killing version, so it will do the edits and then kind of edit the scissor itself and then die. So yes, there are absolutely controls that we can put in place. The one maybe caveat to that is even populations in labs, so just simulated populations, have often developed resistance very quickly. So if you have one mosquito there that has an alteration in the genetic code, that means the scissor cannot recognize and cut. That is quickly going to survive and pass it on. So it's maybe not quite as much rainbows and unicorns as I sold it as first.
Thank you. The next question comes from Carol, and it has a little bit of background. So the question is, 60 years ago, Dr. Leroy Augustine, an MSU professor of biophysics, tried to educate his colleagues and others about the science and moral decisions through a series of television programs on WKAR, as well as a book called Come Let Us Play God, as president of AAAS. His brother had Down syndrome. Should we, he asked, eliminate the genetic anomaly that altered his brother? Shouldn't scientists participate in developing the public policy that allows gene manipulation? Who might encourage scientists to engage in the public square?
Ooh. That's a wonderfully thoughtful question that I'm totally not going to answer. Essentially, because I hesitate to speak for an entire community of scientists. I can give you my personal perspective. I think it is on us as educators who are passionate about this field to encourage that type of communication. So in my classes, whether I'm teaching introductory biology or really anything else, a major focus is science communication. And I do agree with you that we have maybe a little bit of a breakdown in communication between the scientific field and maybe our getting our messages effectively communicated. And maybe part of that is because often, at least me thinking back to my undergrad, I had some communication classes, but not a whole lot. And so it was a lot of self-learning. And I think this is why at least the places that I teach at, like Briggs, are so very hopeful to me, because we have historians and philosophers and sociologists who really think about science in these really different ways. And they have a very, very powerful impact on the ways that our students think and interact and communicate. So I would say yes to all of what you just said. Let's do more of that.
Thank you. Really interesting response from the perspective of an educator. This comes from Bennett, and his question is, in gene therapy, how is a particular type of cell targeted for correction?
Oh, that is such a good question. And that is actually so once you get the CRISPR-Cas9 into a cell, the edit is incredibly efficient and easy. The getting it to the correct cell is really our current sticking point. It's why we haven't had a whole bunch of therapies come through. And so the easiest cells to target, if we're talking humans, are things like blood cells, because you can straight up just take them out of the body, edit them in isolation, and put them back in. An alternative to that may be using cell-specific viral coats. So if you have a virus that you know will only infect breast cells, you can just take off its natural DNA or genetic information and replace that with the genetic information that you want. And it will only take that CRISPR-Cas9 to the breast cells. And so that is one approach that can be used.
I think Judith has her hand raised.
Hi, it's because I don't know how to find the chat box. Okay, what you haven't mentioned here, and which is perhaps should be the scariest point of us all, is genetic warfare, because of what you described could be a huge security issue. And because this may have not only what you do with CRISPR could have unintended consequences, but there's deliberate action as well. If you have one population, an enemy can wipe them out if they can set the CRISPR up to do that. It may take a while to get through the population, but it could happen, couldn't it? And who's looking at that other than the government?
I think given our constraints right now with delivery, like currently right now, maybe not as much of a risk. Technically and theoretically in future, absolutely, it's something that we should be thinking about.
Thank you.
Yeah.
This question comes from Carol, and she wants to know the percent of people who aren't trusting the scientists trying to find treatments or cures for COVID-19 is appalling. Really need to educate. Okay, I guess that's a comment, more of a question, but still an important comment. Dr. Waller has a question.
Yeah. Thank you, Shahnaz. That was so beautifully explained. I have a feeling that the ultimate decisions are going to be made on the basis of money. Who can afford it when it becomes relatively safe to do the germline editing? And because they're bound to find somewhere in the world which is willing to set up a lab where it can take place. But I mean, that's obviously hypothetical. But if that becomes feasible, what do you think are the most questionable edits that could be made? What concerns you most from a moral point of view?
I think that's both a really interesting question and a really hard question to answer. There's no one thing that concerns me. It's really the whole system that we have in place of saying that something is normal, and then anything that we don't define as normal is abnormal and therefore needs to be fixed.
Yeah.
Right? That it's not really one specific thing, but that way of thinking that really concerns me. When as a biologist, I tend to recognize variation in a population as the strength of that population and as the species. But maybe our society and culture and history doesn't really tend to see it in the same way. And so I fear the speed and the access as going down this really dark eugenics path.
All right, we've exhausted the questions in the chat box, but is there anyone who has further questions?
We haven't been very successful, really, in solving many of the problems that we have in this world with any system that is satisfactory to the majority of people. And it seems as though it's going to be even more difficult when we have things like when so many of the countries were facing population explosion and were, allowed to have one child. Sex became an important part. And who decides?
Good question. I don't know. Who does decide?
Dr. Masani, would you mind unsharing your screen? There we go. Now we can see everyone in a full view. Other questions for Dr. Masani? We have one from Nancy. Just how widespread is the distribution of the mail order kit you showed?
That's a good question. I don't really have any specific information for that company. I know that they ship definitely to the US. I know that they also ship internationally. Interestingly, although maybe not surprisingly, the closer there's many places in Europe that have banned this type of gene editing or this type of at least selling the kits without permission. And the closer you get to Germany, in general, the stronger those laws are. I do know that the founder of the company has faced some regulatory battles, but I'm not really up to date on exactly what the legal decisions were. The FDA and the CDC are definitely cracking down. Maybe not quite fast enough, and I worry with the pandemic that maybe they have now other things to worry about, so maybe this is not their priority. Just to be clear, the editing kit that they were selling, it isn't specifically harmful. It's something that isn't going to be harmful, just that specific kit. But once you learn to use those tools, you can make your own DNA for fairly cheap, and that's the worrying bit.
And that's why we need the public discussion of who decides. Are we going to place it in the hands of politicians who are term limited, who at their own desire and whim will call in an expert in one area to say that, no, this particular treatment for COVID-19 is highly desirable and probably effective? You know, who do we believe? Because that's ultimately where decisions get rendered. And I don't know what the current discussion might be in AAAS or other, professional arenas, but I would hope that anybody going into this line of research has at least given it serious thought and is prepared to come to grips with some of those questions.
Absolutely.
Because as you suggested earlier, putting the genie back in the bottle is difficult, if not impossible.
I think you raised a really important point, Carol, about training our scientists not only to be good at the science, but really to be self-critical and to be prepared now to be vocal about these really important issues. We have to put ourselves out there more. We just have to. And we have to maybe talk to each other about what scientific consensus means to us and to others.
Judith, you have a question.
I do. Well, a comment really about how we arrive at getting used to, as a population, getting used to new scientific protocols, things that are possible for us to do. I'm always looking at the broad view, and I'm thinking way back before we had fertility clinics and could use artificial insemination with cattle, which I have done in the past, but we couldn't, if you have a possibility of having, I don't know, let's say cystic fibrosis or Tay-Sachs or any of these other genetic diseases, if you hyperovulate a female and you end up with 5 embryos and 3 of them don't have the condition, and you implant those in the mother, what do you do with the other 2? What is life and what is not life? We haven't even answered that question. So I think if this goes forward and becomes something that is useful to us, that it will be done. I hate to sound like a conspiracy person, I'm certainly not, but without a consensus being reached at all. Because we have actors that are nefarious, shall we say. True? I see you nodding your head.
I think so.
Shahnaz. Yeah.
I think people in positions of power often make decisions without consensus. I'll just leave it at that.
And with at least 51% of them being total idiots, it's probably hopeless. Thank you.
Judith, you also had a question. Schulman. Judith Schulman.
Yeah. Well, I'm sort of a follow-up to both Carol and now I think perhaps Judith. We've got 2 of us here. And as I was listening to the discussion, Am I frozen?
Not yet.
Have I frozen?
No,
No,
Oh, okay. But related to Lyman Briggs, I perhaps am more cynical than most of you. I totally agree with Carol that this is often put in the hands of politicians, the extremely wealthy. But I also am not confident that local communities make decisions that support me, particularly if I'm the, not the 51%. And I would think certainly as a very young adult, where I wanted access to something like birth control. I mean, you have scientific research, and it wasn't just politicians, it was communities in which I lived and around me. So I am less willing to separate us based on only nefarious or only political. I think there are a lot of broader questions about who makes decisions. How do communities that don't have consensus come to agreement? And is consensus good when we're kind of forced to try to reach the best? Anyway, those are my concerns. So I think the politicians are awful, the corporate giants are awful, but I don't have as much faith in our own communities. Anyway, it's also, I find in Briggs, in bringing those kinds of questions within a scientific approach rather than separating them is really beneficial.
good.
Well, we are in Michigan undergoing right now an experiment in public policy. We voted in 2018, if I remember correctly, to establish a citizen redistricting commission, which is now in the throes of trying to develop boundaries for legislative districts. And if we think that that's mundane and not, of importance and significance for us going forward as a state, then, you can sleep through it. But tonight at 7, you're going to be, and this commission is now engaged in some of the nitty gritty, what they can do before they get the census results this year. And that's, part of where the action occurs.
Yeah.
You get a legislature that's predisposed in one direction or another, boundary lines driven by partisan power, regardless of who it is. Are you willing to engage at that level and have a say about communities of interest and other aspects of their work? Because we'll be living with it for 10 years. And that 10 years could make a significant difference.
we've we always have let the Supreme Court of our country make a lot of the moral decisions. And they are currently kind of trying to avoid the one that relates to abortion. And by, happenstance, we now have a conservative 6-3 majority on the Supreme Court. And yet states are, going in each direction, pro and con. And many of those will eventually end up at the Supreme Court. And hopefully we'll have more balance at some time than we have currently.
I hate to interrupt this interesting conversation, but I think Marna has a question.
I do, and this has to do with the medical aspect of gene editing. Is the medical community going in the direction of highest and best use of these tools? For instance, for the most challenging genetic mutations that we have today. And I'm thinking specifically about BRCA1 and BRCA2 that cause breast cancer. And it's 1 in 8 women in this world that get breast cancer. Is there movement in that direction to take action to correct those gene mutations before smaller or less impactful gene mutations are handled?
I think that's a great question, and maybe not one that I'm going to again satisfactorily answer. There is definitely consideration, and there is definitely trials out there. Like there is thought being put into that, specifically with breast cancer and with the BRCA mutations. Again, one big limitation is being able to target the cells. It just isn't as easy to edit anything except, so the reason there's one blood disorders or for the eye is because the eye and the blood are really easy to access and isolate. So the thought and the want is there. I think it's technical hurdles, at least for right now.
Thank you.
Other questions or comments? Here's one from Peter. He's saying that cochlear implants can cure deafness in children, yet some in the deaf community argue that it denies their child the rightful experience as a member of the deaf community. So if you could touch on the ethical questions of curing uniqueness in our communities.
Absolutely. So I think there's multiple layers that we can talk about when we talk about questions like this, right? One is, well, who decides what needs to be treated or cured? That's one. Where do we put our resources, which are finite? Where do we put our funding? What do we back? That's another. And maybe third is, if those therapies are out there, it's not as easy as just saying, well, opt in if you want to. Because then always the issue comes up of, well, what if we're talking about minors? Do minors have autonomy to make their own medical decisions? Specifically with the cochlear implants, which are most effective when the child is younger. These are tough questions that we don't really have answers to.
Thank you. Other questions? Dr. Waller has another question.
Shahnaz, your reference to eugenics got me thinking. It reminded me of a book that was written in the 1990s, I believe, by an American bioethicist called Lee Silver. And I think it was called Remaking Eden. And his argument was that in the future, gene editing is going to allow for the development of two separate classes in society: the gene rich, who come from families who have had all the money to spend on the state-of-the-art genetic manipulation, and they're smarter and fitter and taller and faster than everyone else. And then the gene poor, or the normals, who have not had any benefit from the new gene editing technology. That obviously is based on a lot of assumptions about what is heritable and how many genes are responsible for the inheritance. Do you think there's any plausibility in that? I mean, it's really, what is the H.G. Wells novel, The Time Machine? Do you think that's fundamentally implausible given the complexity of gene expression and gene environment interactions?
I think really this one section from the book I was talking about earlier, The Gene by Siddhartha Mukherjee, he talks about Herman Muller, who is also a famous geneticist, his views on the eugenics movement. Muller was initially, at least, definitely pro-eugenics, and eventually came to think that eugenics and what the original purpose was maybe of the eugenics movement isn't possible without having a society that Mukherjee describes as radically equal. right? So if we inherently have environmental differences, economic differences, all of these differences, then there's no system in which we can decide what's good or bad, or right there, what needs fixing. I think in that way, I think it is definite. I hesitate to say that it's impossible. It can exist. But if you ask me right now, can you engineer a smarter person? I am hesitant to put any stock in that, because it's not about the genes. The ways in which genes and environment interact are so very complex, and our understanding is incredibly small. So, yes, eventually, of course. Is it our primary concern right now? Probably not.
Are there any final questions or comments for Dr. Masani?
Just one comment, if I could.
Yes, Judith.
Can you hear me? Okay. I want to thank you because you explained something to me I did not understand as well as I do now. Not that I don't really understand it, but I have a concept now. And I thank you for your clarity and your time with us today. You must be very busy. So thank you very much. And thank you, John. And thank you, Chris. It's another excellent program.
Yeah, thank you. This was wonderful. I live to talk about what I do. This was such an amazing opportunity. Thanks, Chris and John.
Yes, thank you, Dr. Masani.
Yeah, that was incredibly informative. Thank you so much for it. CRISPR it has such incredible potential, doesn't it? But there are so many troubling paths it might take us down. So thank you so much for making the time. There's time still to enjoy a beautiful evening. But please do come back for Town and Gown next week, when Scott Wolf, who's in the School of Criminal Justice, will be presenting on how we can use science to understand improvements in policing. Hard to get more pertinent than that. So great to see everyone again, and look forward to seeing you all next week. And again, Dr. Masani, thank you so much.
Thank you.
Good night.